Thank you very much. It's not early anymore. Thank you very much. I would like also to thank the organizer, of course, Vice Conti and Paolo Caricetti. I'm not sure if Paolo is already here. I left him in the hotel. He was actually pushing me to come here as early as possible. But so for giving me the opportunity to be here today and share with you a little bit of the things that we have been doing over the last few years. And when Bice told me, look, you are going to talk early in the morning of Saturday, I said Saturday morning over the weekends after the conference dinner on Friday night, so I had to come up with a quite provocative title. And that's why this is Shifting the 100 nanometers paradigm. What is the 100 nanometer paradigm? So you have listened over the last couple of days a presentation regarding the enhanced permeability retention effect, the EPR effect. So these cartoons somehow demonstrate again what you have been listening on the first day and on the second day. And it's the notion that the tumor vasculature is fenestrated with openings, with these large gaps between adjacent endothelial cells. And as compared to the normal vasculature, you can have openings that are as big as 100 nanometers, 500 nanometers, and in some cases also 1 micron. So the notion that has been driving the field of nanomedicine over the last 20 years, I would say, if not more than that, is that if you have a nanoparticle that is spherical in size and is about 100 nanometers in diameter, these particles will continue circulating and eventually will get into the tumor vasculature and passively will cross the fenestration and accumulate into the extracellular matrix within the tumor interstitium. If it is a little bit larger than that size, then the common notion is that it will continue to circulate, get into the liver or into the RES organs and be uptaken by the macrophages and eventually in some way metabolized and excreted if it is biodegradable. And so this has basically, as I said, driven the field and also driven the development of liposomes, polymer micelles, polymeric nanoparticles, and so on. However, as we all know, EPR has some limitations, and this is a very old slide, coming now from a review paper that was published recently, but it's actually a very old research by Rakesh Jain at Harvard Medical School and Massachusetts General Hospital, showing clearly that the size of this fenestration in the tumor vasculature is depending on the type of tumor. And we all know that it's depending on the stage of development of the tumor, it's depending on where the tumor is developing. And it's very interesting to look at this example here where you have two identical cell lines of a mammary carcinoma. One is implanted orthotopically in the breast of the animal, and one is implanted in the back of the animal. Breast, this is orthotopic, this is not orthotopic. And you clearly see that there is a dramatic change in the fenestration size. So we are looking at fenestration that can go all the way from few nanometers up to several microns, depending on where they are developing U87, glioblastoma multiforme, brain tumor. We know that the blood-brain barrier is very tight gap junctions. And even when the tumor is developing, you have sizes that are very small. So relying on the EPR effect, it might be a very good strategy when it comes to small animal models, where you actually inject 1 million cells. In 10 days, you got a tumor size, which is almost 1/10 of the weight of the animal. And everything is, of course, accelerated in an animal model. Of course, we cannot do anything without animal models, but we have to keep in mind that the EPR effect in animal models may be a little bit overestimated. When it comes to humans, we do have EPR effect. Doxil is working in many patients because of the EPR effect, but it might not be as relevant as in animal models. Anyway, even if we have EPR effect and we have fenestrations, this slide and these pictures was already shown by Alberto over the last few days, on the first days, and it comes again from Rakesh Jain lab. You clearly see this is a tumor vascular network, and these are basically liposomes that are stained in red. And you clearly see that there is a red color area in a perivascular position, meaning that the particles are able to extravasate across the fenestrations, but they are not transported deep into the tumor. They are actually stuck immediately in a perivascular position. And you also see that there are a number of side vessels that are not colored at all, meaning that these particles will not get there because maybe there are no fenestration, or because that vascular portion of the tumor is not functional. There is actually no blood flow. And there is another interesting thing regarding what happens in the extracellular matrix. When you do a little bit of engineering and you start measuring the diffusion coefficient of these particles and molecules, here you have the diffusion coefficient of molecules with hydrodynamic radius ranging from 1 nanometers all the way up to our fabulous 100 nanometers liposomes or polymer nanoparticles. And what you see is that compared to a PBS physiological solution, where diffusion in an unconfined space is basically related to the Einstein-Stokes relation, and so it decays with the hydrodynamic radius, as you start moving into the extracellular matrix of tumors that are developing into an animal model, as you pass the 1 nanometer size, you actually see that there is a dramatic decrease in the diffusion. And this is a logarithmic scale, so we're talking of order of magnitude of decay in the transport properties of these particles as soon as you move above the 1 nanometers. So basically you start using nanoparticles and things that are bigger than single molecules or drug molecules. So at the same time, over the, I would say again, the last 2 decades, the notion of targeting the vasculature has been, growing and growing and growing, and has been supported by a number of techniques and experimental data in animal models as well as in human. And this is the work developed by Renata Pasqualini and Wadi Arab, that were formerly at MD Anderson Cancer Center, and now they are at the University of New Mexico, that have been, among with others, developing this phage display library approach, where they can actually map the expression of receptors on the disease endothelium, not only the tumor endothelium, the disease endothelium, cardiovascular disease and neurodegenerative disease and so on. And they actually are able to identify specific receptors as well as motif ligands that will actually recognize specifically those receptors. And it's interesting to see that basically every single disease is somehow associated with a certain level of endothelial abnormality, an overexpression of specific receptors or expression of very specific receptors that are not expressed at all on the healthy endothelium. And so somehow using all these, considering all these, if you want, concepts, the idea that has been driving us has been in developing and rationally designing our nanoparticles has been, rather than that of relying on the EPR effect, is somehow learning from nature and learning from leukocytes and platelets, which are cells that have been designed by nature, I should say, to circulate for a long time, recognize the disease endothelium, stick to the disease endothelium in the case of the platelets, and as well as macrophages. And for the macrophages, of course, there is this very interesting and very unique process of extravasation, which is an active process, and then progressive active migration towards the site of inflammation. And so the design of our nanoparticles and our nanoparticles are somehow biomimicking, as I said, macrophages and platelets. And so they've been selected, if you want, designed to recognize the disease endothelium, land on the disease endothelium, adhere firmly to the disease endothelium, and from there release either your imaging signal or your agent. That now would be a small molecule that can navigate deep into the tissue. So we have particularly my laboratory has been working with 3 different platforms. This is probably the one that also in Italy is more well-known, and it's basically related to the group of Mauro Ferrari. I've been working with Mauro for quite a few years in developing these mesoporous silicon particles, and you will see that there is a reason why they are now discoidal. Originally they were quasi-hemispherical. Anyway, these mesoporous silicon particles, they come in different sizes, in different shapes, cylindrical, spherical, discoidal, they are mesoporous, so that you can load inside these particles through capillary suction a number of things that would eventually stick to the surface on the pores. But there are also other platforms that more recently we have been developing following the same concept. And these are what we call the discoidal polymeric nanoconstructs. They are still discoidal in shape, and you will see the next slides why they have to be discoidal. But they are made out of polymers. They are basically hydrogels. In this case, these are PLGA pegs, so an hydrophobic and hydrophilic chains that are entangled together. And they come in different sizes and shapes, and we use basically a combination of electron beam lithography techniques, so classical microfabrication techniques, or nanofabrication techniques if you want, combined with polymer chemistry and classical polymeric nanoparticle synthesis. You see here some of the steps in the synthesis process, starting from a silicon template and moving into PDMS and PVAs templates, sacrificial PVA templates, and then eventually the individual particles that are, as I said, discoidal in shape, 1,000 nanometers, 1,000 nanometers. So these are conventionally considered very big by the nanomedicine community. Reviewers of papers as well as reviewers of grants, they say these are not nanoparticles, these are micron particles, of course 1,000 nanometers, but it might be true. But so they are 1,000 nanometers by 400, 500 nanometers in thickness. Okay, so they are submicron in size, I prefer to call them, and they can load a lot of things. But we do work also with classical spherical particles, and these are the polymeric lipid hybrid nanoparticles that a group of Bob Langer has been developing over the last 5-6 years together with Omid Farrokhzad. So they have a very classical, traditional configuration. The only thing that is changing is the composition. You have a PLGA core, and also the DPNs have a PLGA core, and they are then coated with this lipid monolayer for stabilizing them, and then of course you have, as always is the case, these peg chains that are sticking outside. So very conventional. The only thing that is changing is somehow the different compartments and the size, again, within 150, 200 nanometers, depending on the payload. Here we are putting inside ultra small superparamagnetic iron oxide. You can decorate the surfaces in this configuration with gadolinium so that you can do T1, T2 MRI imaging. You can do a number of things. I want to emphasize one main difference between this configuration here and this configuration here. We are using the same material. PLGA and PEG are in both systems. But here, PLGA and PEG are entangled together to form a discoidal particles. So we don't have a PLGA core coated by a PEG chains that are sticking out. PLGA and PEG are entangled together to form a monolithic particle. So I guess that beside the size and the shape, there is also quite significant difference in terms of composition, even if taken independently, the ingredients are exactly the same. So why can we do and why do we do rational design? So as you have learned through my the presentation, very kind presentation, thank you anyway, I am an engineer by background. And so for engineers, rational design is something that we have, you know, from our academic background and in our education. And we have been doing rational design on these nanoparticles probably more than 10 years now. And what does it mean? It means combining together computational modeling, so 3 different modules, computational modeling, with in vitro and in vivo experiments. Now, I should say there is nothing unusual in this. It's basically science. You start with the speculation of an hypothesis that may come from computational modeling, and then you do all the steps. You do your validation in vitro and your validation in vivo, right? But these, I should say, in the field of nanomedicine has been quite somehow not used. The field of nanomedicine, to me, is very chaotic and very entropic, if you want to use a a physical term, in the sense that material scientists, bioengineers, engineers like me with very little knowledge of biology have jumped into the field, bringing in their products, and start doing and playing in the labs, generating something and then injecting that directly in vivo, publishing, getting grants, but without really advancing the fields with systematic analysis. So I was very happy when Bice invited me, and very happy also to follow the main talks here during these last 2 days, because you have seen a lot of systematic analysis and a lot of studies in vitro as well as in vivo, showing how products should be actually developed over the years in order to get something that can eventually get into the clinic, or to learn something new, to learn new concepts that can be then used for rationally designing these particles and improving the properties and the outcomes of these systems in vivo. Now, I guess, I present this as the rational design, the integrated framework for the rational design, but really this is a scientific approach that we should all follow in this field. But anyway, the objective of the rational design is that of solving a sort of optimization problem, again, taking advantage of my engineering background. Basically, it's identifying a nanoparticle with a size, with a shape, with the surface properties, and now even with a mechanical stiffness, so the 4S, size, shape, surface, and stiffness, that would actually maximize their accumulation within the tumor or within the diseased tissue, but at the same time minimize their non-specific sequestration by the RES organs, the liver, the spleen, and so on. So a very well-known concept, but we need to do that in a systematic way. And I should say that we can do rational design because we do have access to different platforms. You see the 3 nano platforms that we are using. So we're not working only with one platform that comes from our background, and we know how to play with iron oxide, we know how to play with gold, and so we always do gold, iron oxide, or polymers. No, we are trying to play with different materials, different compositions, different sizes, different shape, and now also trying to modulate the stiffness of our nanoparticles so that we can systematically analyze the effect of these 4 independent parameters. And so, as I said again, we have different platforms that we can use as probes, if you want. And of course, all the different in vitro and in vivo systems that you need in order to do an analysis of the performance in vitro and vivo of the particles. So let me start with the first module, the computational modeling. Of course, I won't go into the details. What is important is to transfer you the message and what we have found. So we have been using these, this is a quite sophisticated finite element model. It's called immersive finite element model. But anyway, what it is a computational model that allows us to model the transport of red blood cells in a capillary, and most importantly, the interactions that nanoparticles will have with these red blood cells and with the flow within the capillary. And so, different snapshots and so on. So what you can do, you can have particles of different sizes. In particular, we have been studying the range between 10 and 1,000 nanometers, and you can answer the question, what does happen to these nanoparticles when they are in a whole blood flow in the presence of red blood cells, considering that the solution that you will inject will get very rapidly diluted into the blood, and so you will have the red blood cells as clearly the most important component in the blood. As you know, hematocrit, up to 40–45% of the volume of your capillary is actually occupied by red blood cells. And so what you do then, you inject these particles with different sizes and you follow their trajectories. And if you do so, and you do it for different sizes, 10, 50, 100, 500, 1,000 nanometers, you immediately realize that for the smaller particles, let's say up to 100 nanometers, these particles will follow what we call the streamlines, will follow the line of the flow. Will deviate a little bit from the flow, but not too much. So if they are sitting next to the wall, they will continue sitting next to the wall. If they are in the core of the channel, somehow mix with the red blood cells, they might drift laterally a little bit, but eventually they will stay in the core of the channel because they are so small that they can find their ways within the red blood cells. If you start playing with some micron-sized particles, 500, 1,000 nanometers, these particles now become in size comparable with the size of red blood cells, which is about 5-7 microns. And so it starts, there is a competition between the two of them. And of course, when you have 1 against 1,000, the competition will be won by the red blood cells. And so what happens is that the red blood cells will push laterally the larger particles. And so the larger particle will continue moving closer and closer to the vessel walls, more than the smaller particles. So liposomes, polymeric particles, 100 nanometers in size with PEG, they are long circulating also because of this. Because it won't be easy for them to get exposed to the vessel walls. It won't be easy to get immediately exposed to the Kupffer cells in the liver that are lining the vessel walls. But at the same time, it won't be easy for them to get to the fenestration in the tumor. It will take some time. That's why you need to have EPR and long circulation with those particles. Those two concepts, they have to go together. You cannot have EPR without long circulation with the polymeric classical or the lipid classical 100 nanometers pegylated particles. These are two concepts that they have to be together, otherwise it won't work, okay? But if you are working with some micron-sized particles, the game is completely different. Now you are already close to the wall. Of course, you need to generate a particle that is able to resist internalization so that as soon as it gets into the tumor, into the liver sinusoid, it doesn't get immediately internalized by the Kupffer cells. But now it's a particle that is moving next to the vessel walls. It's sensing the vessel walls. It's sensing the vessel walls for abnormalities, expression of specific receptors. And by the way, this is what platelets do all the time, searching for vessels that are damaged. And this is what macrophages do all the time on the venular side of our microcirculation. They roll on the vessels looking for abnormalities. They are like sentinels of the vessel walls. And these particles have been designed to do the same thing, sense for the expression of specific receptors, or even sense for the presence of fenestrations that are quite wide to accommodate their size. So it's a completely different approach. Now, theory experiments. In vivo, these are snapshots from intravital microscopy movies showing the distribution in the venules of the ear of a mouse where have been injected systemically 200 nanometers and 1,000 nanometers spherical beads, polystyrene, commercially available, non-pegylated, so very short circulation half-life. But in our case, we don't care about that because we just need to look at the distribution of these particles. And what happens is that is so clear in these images that 200 nanometers particles are quite uniformly distributed across the vessels. So they are, next to the vessel walls, in the middle. The 1 micron beads, most of them are next to the vessel walls, and they are rolling on the vessel walls as macrophages will do. So they are sensing the presence of abnormalities. You also see that the process of, this is a single individual 1 micron bead that is kicked laterally at specific instance from the red blood cells. It's moving longitudinally against the flow, kick laterally, again longitudinal, kick laterally, because at these two time points, there is somehow an interactions with red blood cells, and they are somehow competing and pushing the larger particle out. You can see also in the actual quantification of the distribution for the 200 nanometers is quite uniform. For the 1 micron, it's skewed towards the vessel walls. If you don't believe it, there are also a couple of movies. Let's see if they start, both of them. 200 nanometers, 1 micron. Look at the 1 micron. They are really following the vessels. They are really following the vessels. They are not mixing themselves with the red blood cells. So they are continuously exposed to the vessel walls. In the 200 nanometers case, you see a lot of, let's say, pollution, small particles in the middle, and you see also these larger particles, larger aggregates of 200 nanometers that are actually moving near the vessel walls, because now they are some micron in size. So again, is a different, as I said, is a different approach. Comparison between experiments and theory is quite good. Let me switch back to theory. And now let's get into the hypotheses that we generated with the theory, and then we have been also demonstrating in vitro and vivo. Why do we use these colloidal particles? Now, as I said, spherical particles in a pure laminar flow, which is what you will find in a capillary, in the absence of red blood cells or very near the vessel walls, they follow streamlines, so they will move parallel to their, vessel walls. And so for these particles, it will be quite difficult to interact with the vessel walls unless they are kicked laterally by the red blood cells. But if you start using non-spherical particle, ellipsoidal, discoidal, quasi-hemispherical, you break that symmetry, and these particles start drifting laterally across the vessel walls. And in particular, if they are sufficiently close to the vessel walls, there will be a passive margination motion, taking, of course, advantage of the term that has been used for macrophages, for which these particles will actually drift laterally and get closer and closer to the vessel walls, which is actually what we want. And if you look at the different sizes and the different shapes, actually, the discoidal particle with a very thin aspect ratio, so a very thin discoidal particle, is the one that would marginate and get closer to the vessel walls more than the others. And so this is one of the reasons why we decided 4 years ago to move from quasi-hemispherical to discoidal particle. But there was another concept that we put out back in 2006, where there were only 2 or 3 people, 3 laboratories developing non-spherical particles. Right now there are 5 or 6, no more than that. It's not easy to do non-spherical particles, of course, because you have to use a top-down approach, a microfabrication approach, whereas all the other spherical particles is bottom-up, it's self-assembling, is relatively more simple. Everybody can do that very easily. You don't need sophisticated systems. But the other concept, which for an engineer is very simple, is this. If you have a spherical particle and you want this particle to stick to a vessel walls, remember, and I want to re-emphasize this, whatever I'm saying is for vascular targeting. If you want to target the EPR effect, spherical 100 nanometers is still the best that you can get, and is the way to go. But if you want to target the vasculature, which is a different approach, spherical is not good. So if you want to target the EPR effect, please go ahead with the spherical. If you want to target the vasculature, forget about the sphericals. So if you have a spherical particle that has to stick to a vessel walls, the sphere would have ideally one single point of contact with the endothelial wall, which means one ligand receptor bond, ideally. Of course, it will be a little bit more. But if you have a flat disc, the surface exposed to the wall, will be all the base of the disc. So you will have the chance of generating thousands and thousands of ligand receptor bonds that would actually favor the firm adhesion of this particle to the vessel walls. So it's much more efficient. Not only that, for a given volume, the flat disc will expose a cross-section to the blood flow, which is much thinner than a spherical bead, which means that you will have lower hydrodynamic forces that would tend to dislodge the particle away. So you will have on one side higher strength of adhesion, and on the other side lower adhesion force. That's why in this plot, when you plot the probability of adhesion on a nanoparticle as a function of the volume, and you fix the volume, and then you play with the aspect ratio, gamma equals 1 being a sphere, you see that basically the probability of adhesion grows, grows, and grows as gamma increases, meaning as you move from a sphere to an oblate spheroid that is squished, squished, squished down, and it becomes a limited discoidal particle. And this is again a logarithmic plot, so we're talking of order of magnitude of increase in addition to the vessel walls. And again, this is good for targeting the endothelium. If you want to do EPR, It's not this the approach to follow. And of course, you can use flow chamber system to demonstrate that this is the case. We injected in that flow chamber 3 different particles, quasi-hemispherical, thin discoidal, spherical, and the discoidal were actually sticking to the bottom of the chamber more than the spherical and the quasi-hemispherical. This was also more recently confirmed using the discoidal particles, mesoporous discoidal particles for different shear rates, clearly showing that not only the discoidal are superior to the quasi-hemispherical and to the spherical, but there is, for this shape, an optimal ratio between the diameter and the thickness, and an optimal size, so optimal size and shape combined together in terms of addition to the vessel walls. And if we go back to the theory, you see that each of these parametric curves has a maximum. There is an optimal size. And this is confirmed also in these experiments. And then, of course, we've shown also that discoidal are better than long cylinders. The comparison has been done with the same particle size. And then we move in vivo. Let me do here a trick. We move in vivo, and we injected these discoidal particles into animals, and now here you will see, not really very well, but basically what you should be able to see is, so these are tied to mice where basically the endothelial cells have been genetically modified to express GFP, and so they look green. And then you have in blue the Kupffer cells in the liver. So this is the liver of an animal, abdominal cavity, the animal is alive, is breathing, so you see out of focus movement. This is done with intravital microscopy, and we have been injecting nanoparticles into this system. You see here, there are also bluish elements moving, those are the red blood cells. So the Kupffer cells that you see are basically Kupffer cells that have been internalized in red blood cells that have been stained with a DIO, so a blue dye. And you do see also reddish elements here, which are basically the silicon particles, rhodamine label that are circulating, they've been injected. And we are looking at the liver. Why? Because the liver is, of course, a very interesting organ. No matter what, if your particle is pegylated or not, eventually will get into the liver because they will lose their pegylation and they will get into the liver. So we are not surprised. It's only delaying the lipid peg particles or the polymeric particles will eventually get to the liver. It will take 12 hours, 24 hours, but eventually they will get to the liver, okay? These will get to the liver earlier because they are not pegylated and they are larger, but you know, they all go to the liver. And so basically you don't see much here. Why? Because these are, you know, the first 5 minutes post systemic tail vein injection of the particles. Let's see what happens. Let's see what happens. Xbox, we are not interested right now. Let's see what happens at 30 minutes post injection. Same movie. This is the channel of the nanoparticles. Significant accumulation into the liver after 30 minutes. These particles are not long circulating. We are not, afraid of that. It's a different concept. Co-localization of the silicon particles with the Kupffer cells. Of course, if they get into the liver, they get into the Kupffer cells. That's where all the particles would go. And flow is still flowing. You do see red blood cells here and there. Of course, only 5% of the red blood cells were stained. So again, these are particles that are big, unconventionally big, and they will get to the liver quite significantly, as all the particles would do. But let's see a little bit of quantification with intravital microscopy. Again, this is the setup, the animal, we are exposing the belly, if we need to do the liver here. And look at the accumulation rates of liver and tumor for two differently sized discoidal particles, the 600 by 200 and the 1000 by 400. Interestingly, in the tumor you have a larger accumulation of the larger particles, and in the liver is exactly the opposite. The smaller particles are accumulating more than the larger particles. And you also see that 30 minutes, and this is almost plateauing. So basically no long circulation. But again, if we're targeting the vasculature, and we don't have pegylated particles, we don't need long circulation. We need to get to the tumor right away and do whatever is needed to do. And so, okay, let's see, since those were a little bit controversial as results, again, tumor accumulation for the larger particles more than for the smaller particles, we said, okay, let's take a different, completely different technique in order to quantify the amount of particles accumulating into the liver. And so we use ICP-IS, a quantification chemical analysis of the tissue for silicon, by, of course, subtracting the controls very carefully, because we do have a lot of silicon in our body as well. And what you see is, again, that the 1000 by 400 nanometers are sticking out in terms of tumor accumulation as compared to the larger and the smaller particles. Of course, you do have significant accumulation into the liver and the spleen because these particles are big. But again, these particles are not long circulating, and eventually all the particles will get into the liver and into the spleen. But they do get also significantly into the tumor, even if they are big, okay? The liver to tumor ratio is 3:1, which is not too bad actually considering the size. Then what we did is we conjugated these particles with RGD4C, targeting the integrins on the tumor vasculature. What happens is that you do see across the board an increase in accumulation. Still the 1000 by 400 nanometers, the discoidal, they actually accumulate more than the larger and more than the smaller. And we are getting 8% of the injected dose per gram tissue in this tumor model. We got about the same values also in other, this is a melanoma, we got also similar accumulation in breast and in other tumor models. So these particles, despite their size, they're getting into the tumor vasculature, they are not extravasating, into the tumor vasculature quite consistently, and to values of accumulation which are considerably high. Usually 4 or 5% of the injected dose per gram organ is what you get with particles. With pegylated liposomes, we have seen that you can get up to 10–15% of the injected dose as a peak value, but given the size, this is a very interesting results. And what you see is that basically these particles are landing on the tumor vasculature. They are not crossing the fenestration in the tumor vasculature. They are sitting there, and you can use them for vascular imaging, as well as some molecular imaging of the vasculature if you want, and release whatever you need to get through this fenestration into the tissue. Now, a little bit of analysis of what happens in the liver, again, and spleen. Most of these particles get into the macrophages. They do get there very quickly, but again, this is what happens with all the particles. It's just delaying the accumulation into the liver and the spleen. Now, interestingly, why the larger particles get into the liver less than the smaller, and the opposite happens in the tumor? Because the smaller particle, and by the way, these are still 600 by 200. They are not as small as the 100 nanometers. Anyway, the smaller particles are uptaken by macrophages more rapidly than the larger particles because of their size. And so even in vivo, the Kupffer cells, we take these smaller discoidal particles more rapidly than the larger, and the larger will have more time to get to the tumor and land on the tumor vasculature. Remember that basically the larger particles are more efficient in targeting the tumor vasculature as compared to the smaller particles, which end up into the RES more than the larger. Okay, so now I think it is quite clear why we're using discoidal particles and why we're using some micron. Again, discoidal particles are good for targeting the vasculature. If you want to target the EPR, forget about discoidal. Spherical is basically the way to go. Pegylation is very important for the long circulation. But if you want to target the vasculature, then you need to follow a different approach, and discoidal particles are more efficient. Now, it is true that you have some permeability also in other disease. Even in atherosclerotic plaque, the surface of the plaque is a little bit more permeable. But EPR is really only tumor limited. neurodegenerative disease, cardiovascular disease, where of course nanotechnology and nanomedicine can be of great advantage. You know, probably targeting the vasculature is a more efficient approach rather than targeting an EPR effect, which is not existent in that case. So targeting the vasculature, I believe it might be a more general approach where you can actually slightly modify your system and target the brain vasculature rather than the endothelial cells in the basa vasorum that are developing around an atherosclerotic plaque. So EPR is really only tumor dependent. Of course, if we can solve the problem of cancer, it will be a great breakthrough, but there are many other things that are also very important. So these colloidal particles, this is a new technique that we have been developing and has been inherited by the group of Keenan Park. So these are the hydrogel template strategy. This is an old approach that we have been, as I said, developed from Keenan Park, original paper in 2010, but we have a similar but new technology that is under now protection, and the approach that they are protecting, but the approach is, you know, the objective, final objective is the same. Generate these colloidal particles made out of, in this configuration, PLGA and PEG, but could be any other polymer. But what is important is that we are controlling the size of the shape very carefully. And we're doing that by combining polymer chemistry with a template strategy. So this is a PVA template that eventually you will dissolve in water, and you will get your particles with whatever you have been loading inside. In this case, ultra-small superparamagnetic iron oxide and rhodamine B as a prototype of a drug. And OSPIOs, of course, for doing imaging, and a number of things that I will show in the next steps. So it's a relatively straightforward technique as long as you know how to develop these templates. I should say that dissolving this PVA template is not that trivial as described in Keenan's Park paper, and we have been advancing that quite a lot. And 5 minutes, very good, very good. Okay, so I will run here. So a few examples. These are the particles that are moving Brownian motion. You can see from the top and from the side. They are discoidal, and basically they are quite thin. Of course, they are not perfectly discoidal as the silicon particles. Why? Because these are made out of polymers. They are deformable. They tend to swell also when they are in a liquid. And this is something that we like, because it does allow us to modulate the fourth parameter, the stiffness, which we think is going to be very important for these size of particles. Okay, other characterization. Of course, if they are loaded with ultra-small superparamagnetic iron oxide, they can become magnetic when exposed to an external magnetic field. And in particular, here you see 5 seconds, 10 minutes, you see that basically in the presence of a magnet, these particles are all moved on the side. And I'll get back to this. Other examples showing the progressive degradation of these particles, cytotoxicity, of course, PLGA PEG, with the classical NTT assays, you would not expect anything different than 100% viability. I'm looking forward to send these particles to Copenhagen and see what happens in terms of complement activation, but I won't be surprised to learn that there is minimal. But I rely on you, Mogimi, for this. And then of course a release of rhodamine B, and you should notice one thing. These particles, they don't get into cells. I'm not going to show here because we are still working on that, but you will see in some of the in vivo data, these particles do not get easily into cells. And this is something that we like very much. Here, other examples, you can co-load a number of things, curcumin and doxorubicin, so a potent anti-cancer drug as well as a potent anti-inflammatory drugs. You see the different colors of the particles. Eventually they get yellowish because dox is red and curcumin is green. And you see they are sitting next to the cell. This is not next to the nucleus. And they are releasing their content and then eventually get into the cell. But let me go back to the ultra-small superparamagnetic iron oxide. Huge doses of iron that you can load per single particle, huge doses. So we were talking of stem cell tracking with MRI. Now we have huge doses of iron that can be delivered to the particle. Of course, getting this particle into cells is difficult. Getting particles into stem cells is even more difficult, but it's something that's doable, even attaching the particle to the cell. So you can load a lot of things in these particles because they are big, and they can carry simultaneously a lot of things. But what is more interesting is that in terms of relaxivity, so MRI properties, you start with OSPIOs that are 100 millimolar sec-1 in relaxivity, which is a way to measure the contrast enhancement that can be generated from that particle. The same nanoparticles, 5 nanometers, loaded into these hydrogel-based nanoparticles, they give a signal which is at least 8 times larger. So there is a massive enhancement, a huge concentration of iron, massive enhancement in MRI properties, an additional characterization, modeling, why is this happening? We have shown also that this enhancement in relaxivity is happening also for gadolinium in the past. But one thing that is also very interesting is that now that you can load all this iron into these particles, you can actually do very efficiently magnetic dragging. You know that magnetic forces are proportional to the volume, and so nanomedicine and nanotechnology is not really fan of magnetic forces. They go down, you go down with size, surface forces are enhanced, volume forces are reduced. But now if you have multiple 5 nanometers particles in a large particles, you can actually use these magnetic forces for driving and guiding these particles wherever you want, even deep into the tissue. Why? Because now you have a lot of iron. A lot of iron made out of 5 nanometers OSPIOs that in 1 week, 2 weeks will be completely degraded. So you have both functions simultaneously. You have the biodegradation of the system as well as the magnetic properties that are still there and can be used as a collective effect. And this only can happen with larger particles. I won't go into these details. Here, MRI. Free SPIOs loaded into the particles with a magnet on the side of the tumor, massive accumulation in this case, nothing is happening here. Let me jump right away to the last 2 slides that maybe will generate some comments. These are the DPNs tagged with copper-64, and these are biodistribution, well, not even biodistribution, these are circulation analysis into animals that are free of any disease, no tumor at all. 20 hours, 44 hours, okay? So you see a lot of signal here. This is free copper, by the way, the one that you can use in the clinic, and these are the copper DPNs. You see a lot of signal here. These are basically normalized by the activity, so this is directly comparable with this activity. And you see at 20 hours that you have much more activity associated with the DPNs than for free copper. Nothing surprising, free copper is a circulation half-life of a few hours, of course, right? It's a small molecule. You see a lot in the liver, but they are circulating, because if you see all the signal also on the side and from the top on the transversal view, these particles are still circulating. And you actually get almost about 20 hours of circulation half-life with these particles. 1 micron by 400 nanometers, 1 micron by 400 nanometers, polymeric particles. So they do circulate quite a lot. But let's look what happens into the tumor over time. 1 hour, 6 hours, 20 hours. This is the tumor. Progressive accumulation of these particles that are still big. These are the classical small pegylated particles. They're also circulating. These activities can be clearly compared. They have been normalized. There is some accumulation in the tumor, but not much. They get to values of the order of 3–4% of the injected dose per gram tissue, which is quite normal for these particles. Here, this is one experiment, very preliminary, but the circulation half-life is telling us that these particles are circulating, and so they would tend to accumulate more and more into the tumor. We are getting around 10%, let's say 10% of the injected dose per gram tissue. But what is even more interesting is that the liver to tumor ratio is now getting into the 0.5. And as I told you, these particles do not get very easily into cells, including macrophages. We have light cell microscopy studies with J774, bone marrow-derived monocytes, so primary cells as well as cell lines, showing this particle dancing around the macrophage for a couple of hours without being uptaken, without stimulating the monocytes. We have side comparison with polystyrene particles. All the polystyrene particles are inside the macrophages. These are still dancing around. And I think it has to do with some of the properties that I've described before. This is very preliminary. I just want to let you know while we are working on this. It seems very encouraging, and I'm looking forward to your comments. That's pretty much it. I'm not going to go to the conclusions. And a number of people, of course, in my group that have been helping developing this strategy, and of course, acknowledgement of all the financial support, including the Cancer Prevention Research Institute of Texas. I hope it was quite provocative, and that was my intention as being early morning on Saturday, and I'm looking forward to questions. If there are no questions.
Thank you, Paolo, for your lecture.
Yes. It was so provocative that everybody was messed up.
I will try to be more provocative than you. Why we are studying so basic science in drug delivery in 2013? Where we
because there has been a lot of non-systematic, no, I understand, but there has been, in my opinion, over the last 20 years, there has been a rush to get grants and a rush to publish that has actually killed the field. As I said at the beginning, I believe that many people like me, I'm a biomedical mechanical engineer by background, so I'm not a chemist, I'm not a pharmacologist. But many people like me have jumped into this field, bringing their own technology and trying to push their own technology as much as possible. That's why we're working with 3 different platforms. That's why we're working with 3 different platforms, because we like to do systematic analysis. Unfortunately, systematic analysis don't get high impact factor journals. I had to add the carbon nanotubes to get a Nature Nanotech paper. But I think they are very important in this field.
Oh, no.
Well, they are very important in every field, of course. But absolutely. I think there has been a very chaotic development in this field, to the point that the